Hello, my name is Marcelo Serna, and this is my video presenting the challenge for in vitro experimentation. So, okay, introduction, City Roots Farm, which is a company that does microgreens, has encountered some problems with fungal contamination in their harvest, especially in coriander. So we were tasked to find a solution to this through plant biotechnology. So we found neem. Neem is a biofungicide, it's a natural herb that has biofungicidal properties due to some compounds in its, in it. This is some background information about neem and the effects it has. So we decided to do an experiment to test its biofungicidal effect on coriander. The hypothesis was that this experiment is that the neem extract will have an effect on the growth of contaminating fungi in coriander microgreen cultivations. We designed an experiment in which the independent variables were the type of treatment, the source of treatment, the concentration and the time of cultivation. The response variables were stem length, percentage of germination, percentage of contamination, and wet biomass yield. And the controlled variables were amount and type of substrate, amount of water, and light conditions, and temperature. So for the microgreen experiment, we determined through bibliographical research that the ideal concentration of neem extract for coriander is 5%. So since we were assigned the low concentration, we did 2.5%. We then did an emulsion and diluted with water to have 100 milliliter 2.5 neem extract solution. We had some trays, which were cut. We applied peat moss as a substrate. We counted per tray 2 grams of coriander seeds, which counted them individually for germination percentage. We planted the seeds. We applied the neem extract treatment and some water, and we left them to grow for 15 days at room temperature. Every few days, we took germination percentage data and applied some water. And on day 15, we measured final average stem length, wet biomass yield, germination percentage, and contamination percentage, which was 0%. No trays were contaminated. We also did a cytotoxicity test for neem extract. We once again made an emulsion and diluted it to a 4 milliliter 5% neem extract solution. We kryptonized some cell lines, which were C2, C12, and CACO2. We did the procedure in order to apply them to a 96 well plaque with our neem extract solution. And following Promega's cytotoxicity assay, we were able to determine the cell lines' cell viability with 5% neem extract. Well, the results. Here are the results for average stem length on microgreens per tray. Here are the wet biomass yields per tray in grams. Here is the average percent of germination depending on the day after seeding. These are neem and this is control. And these are the results. We did statistical analysis for the length, the average stem length. We did a NOVA test. This is the null hypothesis that the concentration of neem extract does not have a significant effect on the stem length. The alternative is that it does. And we obtained a p-value higher than an alpha value, so we failed to reject the null hypothesis. So it doesn't have an effect on average stem length. We also did an ANOVA test for germination. We did 2 factors, which were concentration of neem extract and days after seeding. Once again, the null hypothesis is that these do not have an effect on germination percentage. The alternative is that at least one of these factors does. Now, for both factors, we obtained a p-value lower than our alpha value, so we reject our null hypothesis. So both factors have an effect on the percentage of germination of coriander. These are the results for cell viability. We had to reject the results for CACO2 cells because it seems to have been contaminated or there was some mistake. We're not sure what happened really, but we had to discard the data for CACO2. So on C2, C12, we had an average cell viability of 93.84%. The discussion, we compared the results we obtained with literature, and they seem to somewhat coincide with what we should obtain. We obtained 0% fungal growth, which is good, and that's what should have happened. So in conclusion, the concentration of neem extract does not have an effect on average stem length, but it does have an effect on germination percentage and fungi growth. It was a pretty successful experiment. So we could recommend to City Roots Farm to use neem extract to solve the problem they had. And finally, we had some biotechnological techniques that were innovative, such as micropropagation and the use of biofungicides. It also has some market value, I guess, because now City Roots Farm can use the results of this experiment for their advantage. Some advantages of our experiment were that we did provide a very useful solution for City Roots Farms. Some disadvantages is that the scope of our experiment, of Team 4's experiment, was pretty low since we only did 1 treatment with 1 concentration. So it would have been interesting to do more concentrations or different kinds of treatments. But yeah, that's kind of it. Thank you.